Namaste friends, welcome back to Biology Center for True Learning. In this video, we talk of another concept from the genetics chapter, that is backcross. Backcross. What is backcross? Cross the F1 hybrid plant with any of its parents. Crossing of F1 hybrid plant with either of its parents, like a dominant parent or recessive parent, is called a backcross. Backcross is of two types. So one type is dominant backcross. The second type is recessive backcross. Dominant backcross and recessive backcross are types of backcross. Here, dominant backcross is also called outcross. The another name for dominant backcross is called outcross. The another name for recessive backcross is called test cross. In this video, our focus mainly on the test cross. So anyhow, I tell you briefly about outcross and then go for the explanation of test cross. So what is outcross? Cross the F1 hybrid with the dominant parent plant is called a F1. It means outcross is equals to F1 hybrid into dominant parent. So it is the formula for remembering the outcross. So what is the main purpose of outcross? So outcross can be used to improve the quality of breed. Improve the quality of breed. Right? Next, come to the another type of backcross. It's called test cross. It is opposite to the outcross. So what is test cross? Cross the F1 hybrid with the pure recessive parent plant. Recessive parent plant. Then test cross is equals to F1 hybrid into recessive parent. So it is a formula to remember the test cross. Here the experimental parent is same, F1 hybrid. Then the parents are different. In case of outcross, we take dominant parent. In case of test cross, we take a recessive parent. So why test cross is to be performed? So what is the necessity? Generally, in F2 generation of monohybrid cross and dihybrid cross, so we got plants with dominant character, so with different genotypes. For example, so in the F2 generation of monohybrid cross, out of four, 3 plants are tall. Three plants are tall. So among three, so which plants are homozygous plants, which plants are heterozygous plants? So it is impossible to tell just by looking externally. To find the exact genotype of those three plants in the F2 generation, the Mendel was developed the test cross technique. So who developed the test cross technique? Mendel. By Mendel it was. So what is the purpose of test cross? Determine the genotype of unknown individual. So in the crosses, we don't know the genotype of individuals in the F2 generation. To find that genotype, so we can use the test cross tool. So let us understand so this cross. So with the examples, the first we apply this technique for the monohybrid cross, then go for dihybrid cross also. Application of test cross for monohybrid cross. Let me first write parent generation, capital T, dominant tall, small t, recessive dwarf plant. So when we make cross between these two plants, so all the plants we obtained in the F1 generation are heterozygous tall plants. It represents the first filial generation of F1 generation. Then we go for selfing. The cross between the F1 generation plants. The capital T small t into capital T small t, right? The gametes produced by these plants have, half of them have capital T allele, half of them have small t allele. Same here also, half of the gametes have capital T allele, half of the gametes have small t allele. Then go for the crossing. So in two ways we can make it crossing, just with the help of lines and also by the using the Pinner square. Here I directly make the crossing. So this gamete have possibility to fuse with this gamete, then the offspring will be capital T small t. Then again this gamete also have chance to fuse with that gamete, the offspring will be capital T small t, that is heterozygous tall plant. Then so this gamete have chance to fuse with this gamete, the plant will be small t, that is a homozygous recessive. Then this gamete also have chance to fuse with this one, the offspring will be small t. So here we need to observe the results. So what happened here? The two plants out of four, these two plants are same. These two plants are heterozygous tall, right? These plants are heterozygous plant. These two plants are homozygous dwarf, I mean short plant. Here the resulting offsprings will be in the ratio of 1:1. So what is the ratio we obtained by the test cross in the monohybrid experiments? 1:1. Because so 1/4, 1/4, 1/4, 1/4. 1/4 plus 1/4, 1/2. So 1/4 plus 1/4, so 1/2. 1/2 plus 1/2 is equal to half. So 1:1 ratio. So this is the, when we get this ratio, so what we should understand is the experimental plant. So we take in the experimental plant. So we take here is the homozygous plant, right? So let me do some correction here, right? Actually here, so we do the cross with the small t actually, right? So in test cross, the F1 hybrid so will be crossed with the recessive parent. So these are the gametes. Then finally, so these are the offsprings we got. The ratio we obtained in the monohybrid cross, so by test cross is 1:1. For suppose the experimental plant, right? This is the experimental plant, right? When experimental plant is a heterozygous plant, so this is the ratio we obtain. In case of experimental plant is a homozygous plant, so we don't get this one. So we also check it here. If the experimental plant is homozygous, then cross it with this recessive plant, homozygous recessive plant. All the plants we obtain capital T, small t, then capital T, small t, then capital T, small t, then capital T, then small t. All the plants are tall. All the plants are tall. The 100% the offsprings we received here are tall plants. So here we did not obtain the 1:1 ratio. By this result, so we can conclude that the experimental plant taken was homozygous tall plant. Now we go for apply the test cross tool for dihybrid cross also. So how to find the genotype of unknown offsprings in the F2 generation of dihybrid cross? Dihybrid cross. Here parents capital R, capital Y, capital Y, then small r, small y, small y. So when we do cross here, the offsprings we obtained in the F1 generation have the genotype like this: capital R, capital Y, small y. This is the F1 generation. Now what we need to do under the test cross? The cross this F1 hybrid with this recessive parent. First, to make the process easy, we need to write the gametes. This F1 hybrid can produce 4 types of gametes, like capital R, capital Y, then capital R, small y, then small r, capital Y, then small r, small y. These are the 4 different gametes produced by the F1 hybrid in the test class. Then what about this one? So all the gametes produced by this plant have small r, small y allele. All the gametes produced by this recessive parent plants have small r, small y capital. Right down. Actually, this parent plant express a dominant character round yellow color seeds. This plant express wrinkled green color seeds. Right. So now we go for so draw the checkerboard to know the results. So draw the 16 squares, inner square, 1, 2, 3, 1, 2, 3. So take the gametes of the F1 hybrid vertically, RY, small r, capital Y, small r, small y. Then write the gametes of recessive parent horizontally, RY, all gametes containing of small r, small y alleles. Right. Now do the cross. Capital R, capital Y, small y. Then capital R, capital Y, small y, capital R, capital Y, small y, capital R, capital Y, small y. Next, the capital R, small y, small y. All are are same in this row. Next, here smaller, capital Y, small y, then all are same in this row. Then lastly, smaller, small y, parental type, actually all are same in this row also. Right? So here we need to find the ratio. So let me erase this much of part. So in four rows or in four columns, four types of plants are produced. Four types of plants. Four different types of genotypes are produced. I am writing here them. The first one, capital R, small r, capital Y, small y. The second type, capital R, small y, small y. Then third type, small r, capital Y, small y. Then the last type, small r, small y, small y. First, write the phenotype of these four different genotypes. RRYY means round shaped yellow color seeds producing plants. The phenotype of this plant is round shaped green color seeds producing plant. And phenotype of this plant is wrinkled shaped yellow color seeds producing plant. Then finally wrinkled shaped green color seeds producing plants. Occurrence of this result in the ratio of 1:1:1:1. So here this is the ratio we obtained in the F2 generation by test cross technique, right? So this is about the back cross. Thank you for watching this video.